Assalamualaikum warahmatullahi wabarakatuh. My name is Siti Nusriani binti Hussein with metric number 206079. Today, I'm going to show you a step-by-step technique of streaking for single isolated colonies. The streaking technique that I'll be performing in this video is streak plate method. The streak plate method is a rapid qualitative isolation method that is used to separate the cells of a mixed culture so that discrete colonies can be isolated. The main idea is that the sample or culture will be diluted when it is spread over the surface of an agar plate. So, this will result in the decreasing number of organisms present from one quadrant to another. By third or fourth quadrant, only a few organisms will be present. The decreasing number of population size ensure that, after inoculation, individual cells will be sufficiently far apart from one another on the surface of the agar medium. The materials needed for this experiment are a Bunsen burner, nutrient agar plate, bacterial culture, inoculating loop, and a marker. First and foremost, using a marker, draw two bisecting lines on the bottom of the petri dish to divide the plate into 4 equal parts. Label each quadrant 1 through 4, starting with the top right quadrant and labeling counterclockwise. Turn the petri dish over and then flame the loop and cool it down for several seconds. Place a loop full of culture on the agar surface in area 1. Drag it rapidly several times across the surface of area 1. Turn the petri dish 90 degrees. Reflame and cool the loop by touching an unused part of the agar surface close to the periphery of the plate. Then touch the loop to a corner of the culture in area 1 and drag it several times across the agar in area 2. The loop should never enter area 1 again. Then turn the dish 90 degrees, reflame and cool the loop. Streak area 3 in the same manner as area 2. Without reflaming the loop, again, turn the dish 90 degrees and then drag the culture from a corner of area 3 across area 4 using a wider streak. Don't let the loop touch any of the previously streaked areas. The flaming of the loop at the points indicated is to dilute the culture so that fewer organisms are streaked in each area, resulting in the final desired separation. As a result, Quadrant 1, which is area of initial inoculation and the first streak, yield a heavy growth. For Quadrant 2, it yield a less dense growth. For the third quadrant, it yield a weak growth. Lastly, for Quadrant 4, it yield a single colonies or a discrete colonies.